Last time we explained about the different morphologies of the ovarian follicles. We talked about the development of the ovarian follicles from primordial to primary, then to secondary, then to a tertiary, then to a Graafian. We described the morphological changes, and we talked a little bit about how this pre-entral, we said that we have pre-entral follicles and enteral follicles. When we have the appearance of this entrum, of this cavity containing a fluid inside of the ovarian follicle, we start to call the follicles as enteral follicles. And the stages before, where we don't have an entrum, the follicles are called pre-entral follicles. We said that the development of the follicles from primordial to pre-entral, to primary and secondary follicles, is independent from the gonadotropins, is independent from the action of the LH and the FSH. And it's the late secondary follicles, so the late pre-entral follicles will acquire FSH and LH receptors, and the FSH receptors will be expressed on the granulosa cells, and the LH receptors will be expressed on the theca interna cells. Okay. Now, the secondary follicles will develop to give enteral follicle. This development of the secondary follicles, those are the secondary follicles into enteral follicles, can occur under the action of FSH because they start to express FSH receptors now. Those secondary follicles, they can respond to the action of FSH, but they can develop also to give enteral follicles independently of the FSH. Under the action of intra-ovarian factors, so under the action of some cytokines produced by the follicle itself. Okay, so this development from the secondary follicles to the enteral follicles can be independent from the action of FSH, but can also occur. It can also occur under the action of FSH. Those secondary follicles will give early enteral follicles or tertiary follicles. Those early enteral follicles now they express. Since we have them, we will have a pool of early enteral follicles produced, and those early enteral follicles are going to develop to give late enteral follicles or Graafian follicle or a pre-ovulatory follicle. Okay, so we will have a transition from the early enteral follicles to the pre-ovulatory or Graafian follicle, and this transition from an early enteral follicle, from the early enteral follicles into the pre-ovulatory or Graafian follicles, occurs in 3 stages. First of all, we will have the recruitment process, then we have the selection and the dominance process. Okay, so we have 3 stages of this transition from the enteral to the pre-ovulatory follicle, and those stages are first the recruitment, then the selection, and the dominance. So those early enteral follicles, many of them at the beginning of each menstrual cycle will be, they have now receptors for the FSH and the LH, okay? And under the action of FSH, here we have the endocrine or the gonadotropin, the variation of the gonadotropins during the menstrual cycle, and the variation of the estrogen and progesterone during the ovarian cycle. At the beginning of each menstrual cycle, we have those follicles, we have high levels of FSH, And since those follicles here, they express FSH receptors, they can respond to the action of the FSH. Okay. So, under the action of FSH, a pool of early enteral follicles will develop, will start to develop and to get a little bit bigger, to increase in size. But not all of this pool, not all of them, they are not synchronized in their development. Some follicles will be bigger than others. Okay. And among those developing follicles, so those enteral follicles that engaged their development, which will engage their development, will say they are recruited, and this is the recruitment process. So at the beginning of each menstrual cycle, under the action of FSH, a pool of early enteral follicles will be recruited to develop into more mature follicles. Okay, and as I said, not all the development of those follicles is not synchronized, so not all of the follicles will have the same size at the same moment. Some of them will be bigger than others, and others will be smaller than the others. But among all of those recruited enteral follicles and developing enteral follicles, one of them will be more important in size than the others. Okay. Now, the reasons why this one will be more important in size are not very well understood, but the hypothesis is that this follicle, which is bigger than the others, has more FSH receptors on its granulosa cells and can respond better to the action of FSH and increase faster in size. So we will have like a leader follicle, which is bigger than the others, and the others which are smaller. And this bigger one will be the dominant. We will call it later the dominant follicle. Okay, so we will have one bigger follicle that we will call it the dominant follicle, and others that are smaller. Now, those developing follicles, under the action of FSH, they can, in fact, under the action of the if there is LH, the theca cells, since they have LH receptors, can produce androgens, and the granulosa cells, under the action of FSH, will synthesize the aromatase, which is the enzyme that will convert the androgen into estrogen. So due to this action of the FSH, the granulosa cells of those follicles can start to produce estrogens. Okay, before the enteral stage, the the granulosa cells didn't use to produce estrogens. Okay, now the recruited follicles, they start to produce estrogens by the granulosa cells, and this is due to the action of the FSH, as you can see here. Due to the production of under the action of the FSH, those follicles, now they are small, they produce very low levels of estrogen, but when we will have the dominant follicle, the big one bigger than the others, this dominant follicle will produce more estrogen levels than the others, and this will lead to a little increase in the levels of the estrogen between the 5th and the 8th day of the menstrual cycle. And so we will have low levels of estrogen secreted in the blood. And those low levels of estrogen, so the estrogen secreted will have a negative feedback, will exert a negative feedback on the GnRH secretion by the hypothalamus and on the FSH and LH secretion by and on the pituitary gland, which will lead to an inhibition of the FSH production. Okay, and also under the action of FSH, those granulosa cells, we said that they produce estrogen, but they will also produce inhibin, and the inhibin also exert a negative feedback on the anterior pituitary gland, and as a response, this anterior pituitary gland will stop to produce FSH, or the levels of FSH will drop, will decrease markedly. So we will have a drop due to those to the estrogen secretion and the inhibin secretion by the granulosa cells. We will have a negative feedback on the FSH production by the anterior pituitary gland. And this will lead, so this dominant follicle is producing estrogen. Now, the levels are a little bit low, but at this low concentration of estrogens, plus they are producing inhibin, we will have a negative feedback on the production of FSH. Now, we will have a drop in the FSH level, as you can see that starting the second week of the menstrual cycle, the levels of FSH will drop due to a small increase in the levels of estrogen. Okay. Now, those follicles were dependent in their growth from the FSH, because the FSH, if you remember, we said this during the last session, the FSH will lead to the division of the granulosa cells and the increase in size of those follicles. And that's why those enteral follicles, which can respond to the FSH, once recruited, started to increase in size under the action of FSH. Okay, they started to develop under the action of FSH to become bigger. So the development of those follicles is dependent from the action of FSH. Now that we have a drop in the levels of the FSH, what will happen to those follicles? In fact, all of them will die except for this dominant follicle. Only the dominant follicle, the biggest in size, can survive, but the others will die. And this is because the levels of FSH produced at this moment are lower than their we need some threshold, okay, to have a growth or a development of those follicles. If the levels are below this threshold, those follicles cannot survive. However, the biggest one, the dominant one, has more receptors of FSH and is more sensitive to the FSH, and it can bind more FSH molecules because it has more receptors, and it can keep on surviving in the presence of those low levels of FSH. So due to this drop in the FSH levels, only this biggest one, which will be the dominant one, will keep on surviving, and the other follicles will die. And so this drop of the FSH level will lead to what we call a selection of a dominant follicle. So due to this FSH only one of the recruited follicles will be selected. The others will die by atresia. And here we can say that we have one selected follicle, which is the dominant follicle. Okay, and only this dominant follicle can keep on increasing in size and developing to give the pre-ovulatory follicle, and the others will die. Okay, so only one enteral follicle, the dominant one, will be selected to continue the follicular growth. The others will degenerate or will die by atresia. And in the absence of FSH, in fact, this dominant follicle, the estrogen also can help in the survival and the development and the growth of those follicles. So this follicle can grow due to the presence of estrogen, because the estrogen can exert a positive, can exert a paracrine and autocrine, can act in a paracrine and autocrine manner on the granulosa cells themselves and to induce their proliferation. So the estrogen also affects the growth of the follicles. So this follicle can grow, and also due to the presence of LH, because now those dominant follicle, or this dominant follicle, before its selection, under those follicles recruited under the action of FSH will start expressing on their granulosa cells LH receptors. So now the presence of the LH will help also this dominant follicle in its survival, and it can keep, so it can keep on surviving and developing even in the presence of low levels of FSH. Now here we can see that, so at the beginning of the menstrual cycle, we have high levels of FSH. Those FSH, now at the beginning of each menstrual cycle, we have early enteral follicles that are present, and they will be recruited to develop under the action of FSH. Since we have high levels of FSH, those early enteral follicles can be recruited to develop and to give more mature follicles. Those more mature follicles, under the action of FSH also, their granulosa cells can start to produce estrogen. And by the end of the first week, and by the 5th to 7th week, we'll start having little increases in the levels of the estrogen in the blood due to the secretion of estrogen by the granulosa cells. And those estrogens levels plus production of inhibin also by the granulosa cells will lead to a negative feedback on the FSH production, which will lead to a drop in the levels of the FSH. And then we will, due to this drop in the FSH levels, we will have a selection of a dominant follicle. Only one large enteral follicle is selected. This will be the dominant follicle. And as a consequence, the others will die, the other follicles will die, and only this dominant follicle can keep on surviving and growing to give the pre-ovulatory follicle. Now, this, is it clear till now? واضح لكم؟
Yes, doctor.
Okay. The dominant follicle produces estrogen, and it increases during its development. It will increase in size, so it will have more granulosa cells, will have more estrogen production, and also due to the action of FSH, they start to express FSH LH receptors on their granulosa cells, because as you know, in the early enteral follicles, we have LH receptors only on the theca interna cells, but the granulosa cells don't express LH receptors. Now, while maturing, this dominant follicle will acquire more LH receptors on its granulosa cells, and at certain moment, this dominant follicle, as we said, will become important in size. It has a lot of granulosa cells. All of those granulosa cells are producing estrogen. We will have high levels of estrogens in the blood. So this large follicle now, the dominant follicle, because it is big in size, can produce large amount of estrogens, which will lead to a peak of estrogen a little bit before the ovulation, okay, 2 days, almost 2 days before the ovulation. Okay, now the ovulation, normally the menstrual cycle lasts for 28 days and the ovulation occurs at the 14th day. And the phase before the ovulation is called the follicular phase, and the phase after the ovulation is called the luteal phase. Okay, so here we have the follicular phase where we have the follicular development, then we have the ovulation, then we have the luteal phase that we will talk about it later. Now, we are still in the follicular phase here. The dominant follicle is increasing in size and producing more estrogen. We will arrive a little bit before the ovulation, and due to those levels of estrogens produced by the dominant follicle, we will have a peak of estrogen. And those high levels of estrogens produced by the dominant follicle now, they exert the large amount of estrogens produced by the dominant follicle will have a positive feedback on the hypothalamus and on the anterior pituitary gland, and more specifically on the secretion of LH by the anterior pituitary gland. Okay, so due to those large amount of estrogen, we will have a positive feedback on the hypothalamus and on the anterior pituitary gland that will lead to an LH surge. So we will have a peak of LH. We can see here that directly after this peak of estrogen, we have a peak of LH at the day 13 before the ovulation. We have also a peak of FSH because we have a positive feedback on the anterior pituitary gland and the hypothalamus. But this peak of LH will have very important consequences on the further steps of the folliculogenesis and the ovulation, and for the later stages of development. This LH peak, in fact, will be responsible for the ovulation and will be responsible for the production of progesterone by the granulosa cells. Okay, we will talk about them in details now, but it is due to this LH peak that now this dominant follicle will, under the action of LH, will stop producing estrogen and they will start producing progesterone, and also we will have the ovulation. And if you remember also, we have said that under the action of the LH, the primary oocyte will resume its meiosis and will achieve the meiosis 1 and enter in the meiosis 2. All of those these things are consequences of this LH peak. Okay, so the dominant follicle here produces large amounts of estrogen, and those large amounts of estrogens, they have a positive feedback on the hypothalamus and on the anterior pituitary gland, which will lead to a peak or a surge in the LH levels. And this surge of the LH will lead to the ovulation and to many changes in the follicle itself. Okay, and in the oocyte, at the level of the oocyte, like the resumption of the meiosis, for example. So at the beginning of each menstrual cycle, we have early enteral follicles, which are tertiary follicles that will develop to give late enteral follicles. And in fact, all of those steps of follicular development, they occur in this first part of the menstrual cycle, in the follicular phase of the menstrual cycle before the ovulation. After the ovulation, we don't have any follicular development because the follicle reaches its maximal development before the ovulation. Then we will have the ovulation. Okay. After the ovulation, we will talk about the luteal phase. So all of those steps of follicular development will occur here in the first 14 days of the menstrual cycle before the ovulation. And this transition from an early enteral follicle to a late enteral follicle occurs under the action of FSH and estrogens, because the estradiol is an estrogen. Okay, we have two major estrogens produced by the granulosa cells, which are the estradiol 17 beta and the estrone. So under those changes will occur under the action of FSH that will lead to the development of those early enteral follicles. And the estradiol can also help in the development in the follicles, but also will regulate the FSH levels, because first of all, when secreted at low levels, it have a negative feedback on the FSH productions. Then later on, when produced in large quantities, it has a positive feedback on the hypothalamus and the anterior pituitary gland, which will lead to a peak of LH and FSH. And so in the first week of the menstrual cycle, we have the FSH levels that are elevated and low estradiol levels. As consequences of those high FSH levels and low estradiol levels, we will have the selection of one large enteral follicle, which is the dominant follicle. Now, in the second menstrual, in the second week of the menstrual cycle, the dominant follicle, as we have said, the dominant follicle is almost selected here when we have this drop in the FSH levels by the end of the first week. So in the second week of the menstrual cycle, the dominant follicle will keep on increasing in size. It continues to grow until it reaches the pre-ovulatory stage, or the Graafian, it becomes a Graafian follicle. The other follicles, all of them will undergo atresia due to the drop of this FSH, because we don't have FSH anymore. And the dominant follicle will acquire LH receptors on its granulosa cells, and it will produce large amounts of estrogen. This dominant follicle produces large amount of estrogen that will lead to an LH peak and LH surge. Okay, here also we have the same ideas. We have a cyclic recruitment of early enteral follicles at the beginning of each menstrual cycle, because the development of the enteral from early enteral to late enteral follicles occurs at the beginning of each menstrual cycle and is cyclic, so it depends of the menstrual cycle. However, the stages before the development, the stages before the enteral stage, the stages from the primordial to the pre-enteral follicles, they are not dependent of the menstrual cycle. So they are not dependent, they are not cyclic. We don't have at each menstrual cycle a recruitment of primordial and primary and pre-enteral follicles. In fact, this occurs independently of the menstrual cycles. And as you can see, this pre-enteral stage here is very long. The transition from primordial to primary follicles, it takes 150 days. Then the transition from primary to pre-enteral follicles takes 120 days. And then from pre-enteral to enteral, it takes 70 days. So it's very long, and it's not dependent from the menstrual cycle. However, what is the follicular development, the cyclic follicular development, is the recruitment of the enteral follicles at each menstrual cycle. With the beginning of each menstrual cycle, we will start having a recruitment of a pool of enteral follicles that will develop into pre-ovulatory or Graafian follicles. Okay, and as we have said, this follicular phase from the early enteral to the pre-ovulatory follicles lasts 14 days. It lasts for the first 2 weeks of the menstrual cycle before the ovulation. So the first 2 weeks here, we have this follicular development from the early enteral to the pre-ovulatory stage. And at the beginning of each menstrual cycle, we have those early enteral follicles that are recruited to develop into more mature follicles. So we have cyclic recruitment of early enteral follicles. At the beginning of each menstrual cycle, a cohort of early enteral follicles will begin to develop into large enteral follicles. And this development is under the action of FSH. And because we have high FSH levels at the beginning of the cycles, those early enteral follicles can start to develop, in fact. And the recruitment of a cohort of enteral follicles once in the late luteal phase or early enteral phase of each cycle. So this recruitment of enteral follicles happens in the early follicular phase and at the beginning of the early follicular phase of each cycle, because the follicular phase is called, the early follicular phase is called the late luteal phase. Here we have the luteal phase. At the end of the luteal phase will start the follicular phase. So the early follicular phase is called also the late luteal phase, but we can keep on calling it the early follicular phase. So the recruitment of a cohort of enteral follicles occurs once at each in each early follicular phase of each cycle. Okay, then the selective rise in FSH levels that occurs during the luteal follicular transition. So the selective rise in FSH levels that we have in this early follicular phase, and at the beginning of the menstrual cycle, as we have said, we have high levels of FSH. We have will be those high levels of FSH present at the early follicular phase is a potent stimulus for the follicular recruitment, and several early enteral follicles will begin to enlarge in this phase of the cycle. Now, the future dominant follicle may contain more granulosa cells. Why the dominant follicle can keep, or this biggest follicle can keep on surviving once we will have a drop in the FSH levels, and the other cannot survive? Because the future dominant follicle may contain more granulosa cells and FSH receptors, making it more sensitive to FSH compared with the subordinate follicles, to the other follicles that are less developed. And by the day 5 to 8 of the menstrual cycle, the aromatase activity begins in the granulosa cells of the follicles. The aromatase is the enzyme that convert the androgens like the testosterone into estrogens. Okay, so by the day 5 to 8, we will have the aromatase activity. So those enteral follicles under the action of FSH will start having an active aromatase between the days 5 and 8 and can start to produce estrogen. Okay, and thus starting from now we can start having an increase of the estrogens levels. And in this first phase, after the day 5 and before the peak of estrogen, we have low levels of estrogen that will lead to a decrease in the FSH levels. So by day 5 to 8 of the menstrual cycle, the aromatase activity begins in the granulosa cells of follicles larger than 6 to 8 millimeters, with the dominant follicle producing more estradiol 17 beta than other follicles in the cohort, and the dominant follicle will produce more estrogens than the others. The dominant follicles start to secrete enough estrogen that increases the blood estrogen concentration, and also this dominant follicle secretes inhibin, and the inhibin and the low concentrations of estrogens will exert a negative feedback on the pituitary FSH and LH production, which will lead to FSH levels to decrease. Okay, now the dominant follicle is rich in FSH receptors. It can continue to respond to the low levels of FSH. However, this the FSH concentration that falls below the requirement of the smaller follicles lead to their regression and atresia. So in the presence of those low FSH levels, the smaller follicles will die because this FSH concentration is below their requirement, below the necessary levels for their growth. Okay, and the dominant follicle under the action of estradiol and FSH undergoes a pre-ovulatory growth by developing LH receptors on its granulosa cells. Now, the dominant follicle under the action of FSH and estradiol will develop LH receptors on its granulosa cells. Okay, and thus, the later ovulation can occur in response to the LH surge, because as we have said, the ovulation will occur in the response of an LH surge. So in the LH surge, the granulosa cells will respond to this LH. Okay, so it's like that the FSH and estrogen were preparing those granulosa cells by letting them express the LH receptors. So they were preparing this dominant follicle to respond to this LH surge. Okay. So during the development of this dominant follicle, we will have LH receptors that will be expressed on the granulosa cells. And this will lead to the ovulation later under the action of the LH surge. Okay, is it clear?
Yes, that's
now we can see here the different follicles, different stages of the maturation of follicles. We can see here this is early enteral follicle. This is also no, this is in fact this is a secondary follicle, since it doesn't have antrum, it doesn't have cavity inside it, so it's a late pre-entral, okay, follicle. And this is an early entral or a tertiary follicle, and this is a late entral follicle. The pre-ovulatory follicle will be the Graafian follicle. We can say that this is almost the Graafian follicle, okay? But it's a little bit, the Graafian follicle has a little bit bigger antrum, and here the cumulus ophor is forming like a stalk, okay? So this is an early antral, and this is a late antral follicle. We can see here in the pre-antral follicles, here we have the granulosa cells, and here this is the oocyte, and in blue this is the zona pellucida that separates the follicular cells or the granulosa cells from the oocyte. We can see here that we don't have any FSH receptors at this stage of follicular development. We don't have any FSH receptors, neither LH receptors on the follicular cells. Here in the early antral follicle, we can see that here we have the theca cells, then we have the membrane of Slavjensky, or the basement membrane, called here the membrana granulosa, and here we have the granulosa cells. And the theca at this stage here, because in the late pre-entral, the cells start to express LH receptors on the theca cells and FSH receptors on the granulosa cells. And also in the early antral follicles, we can see that the theca interna cells express LH receptors on their surface that can bind the LH, and also the granulosa cells will express FSH receptors on their surface that can bind the FSH. And in fact, the binding of the FSH to the granulosa cells will lead to the proliferation of those cells, and thus an increase in the size of this follicle. due to an increase in the number of the granulosa cells, but also it will lead to the activation of the aromatase, the enzyme that will convert the testosterone or the androgens into estrogen. So under the action of FSH, the granulosa cells can secrete estrogen, start to produce estrogen. Now in the late enteral follicle, we can see that in the dominant follicle, here has more LH receptors on the theca cells. We still have LH receptors on the theca cells. The granulosa cells have FSH receptors, but also they acquired LH receptors also, okay? And because at the ovulation, they can bind the LH, and this binding to the LH will induce modifications in the oocyte that will resume its meiosis and also will lead to the ovulation, okay, and other modifications at the level of the ovarian follicle that we will see just now, okay. So here we have the same ideas also. Now, what are the changes what are the consequences of this LH surge? We have said that just before the few hours before the ovulation, we will have an LH surge, and this LH surge will have several consequences on the ovarian follicle. Okay, it will lead to the ovulation. It will lead to changes on the follicular cells. Okay. It will lead to changes at the level of the oocyte, okay, and those changes at the level of the follicles and the follicular rupture will lead to the ovulation. So we will start by talking about the changes on the on the follicular cells due to this LH surge, and the effects of this LH surge on the follicular rupture and the ovulation. And at the end, we will talk about the effects of the LH surge at the level of the oocyte. Now, due to this LH surge, we will have a change in the properties of the outer granulosa cells. The granulosa cells will cease dividing because, as we have said, under the action of FSH and estrogen, they were dividing, okay? And the oocyte was increasing in size because more granulosa cells were forming, okay? But now under the action of, and also because the enteral is becoming bigger, but now we will see that under the action of LH, the cells will stop dividing, and the increase in the size of the dominant follicle will be only due to the increase in the volume of the follicular fluid and an increase in the size of the enteral, okay? So under the action of LH, and because the granulosa cells express receptors to the LH, they can respond to the action of the LH, and due to this so to the interaction of the LH with its receptors on the granulosa cells, those cells, the granulosa cells, will stop dividing. They don't convert anymore the androgens to estrogen, but they, however, they will start synthesizing progesterone. Okay, so we don't have any more production of estrogens by the granulosa cells, but those cells, the granulosa cells, will start to synthesize progesterone. And so this LH stimulates the synthesis of progesterone via the newly acquired LH receptors. So this interaction between the LH and its receptors will lead to the activation of the progesterone synthesis. And we will have inhibition of the aromatases, the enzymes that will convert the testosterone into estrogens. That's why we don't have any more estrogens or production of estrogens by the granulosa cells. The granulosa cells will lose their ability to bind estrogens and FSH, okay? And they will gain the ability to bind progestins, progesterons. So those granulosa cells due to this LH surge or under the action of LH, will not be able anymore to bind estrogen and FSH. And also, but they will have the ability to bind the progesterone. And this will result, because this progesterone now, those granulosa cells are producing progesterones and are expressing progesterone receptors in the cells. And the progesterone acts on, in a paracrine and an autocrine way on the granulosa cells. So once it is released by those granulosa cells, it can act on them again and exert a positive feedback on their production. Okay, so this will result in an exponential release of progesterone from the follicle. And the binding of progesterone to its receptors on the granulosa cells will lead to more progesterone production. And that's why we will have an exponential release of progesterone from the follicle, which will become significant in the human several hours before ovulation. So this quantity or those quantities of released progesterones will become significant, important, a few hours before the ovulation. As we can see here, directly after the, as a consequence of the LH peak, or when we started to have the surge of the LH, the progesterone levels started to increase to reach their maximum directly after this LH peak, okay? So the cells produce a lot of progesterone. And this rise in the progesterone levels has three major consequences. It will depress the growth of the less mature developing follicle. If you remember, we said that the FSH will lead to the growth of the less mature follicles, yani early enteral follicles. Now the progesterone will depress or will prevent the growth of the less mature developing follicles. It is essential for the ovulation. This progesterone will activate certain proteases. We will talk about them now. And those proteases will degrade the membrane, will lead to the degradation of the membrane of the ovary and to the exit of the oocyte from the follicle and from the ovary, okay? So it is essential for the ovulation itself, and it promotes the transition to the progestagenic phase of the ovarian cycle. In fact, the progestagenic phase is the luteal phase. So it prepares the transition to this luteal phase where we have a dominance of progesterone during this phase. So those are the three major consequences of the progesterone produced by the granulosa cells under the action of the LH surge, or as a consequence of the LH surge. Now, as we have said, at the pre-ovulatory phase, the follicular cells will start to express LH receptors. The LH action on the follicular cells will induce a follicular growth and activity. And this follicular growth is due mainly to an increase in the size of the antrum due to an expansion of the follicular fluid. Yani the volume of the follicular fluid will increase, and thus the antrum size of this antral cavity will increase, and that's why now we have this increase in the follicular size. So during this, as a consequence of this LH surge, the granulosa cells will not divide anymore, and the dominant follicle will increase in size almost exclusively due to a rapid expansion of the volume of the follicular fluid, because those granulosa cells will release a lot of progesterone and other substances now due to this LH surge. And we will have an increase in this in the volume of this follicular fluid. As you can see, we will see also that due to this LH surge, we will have production of hyaluronic acid by the granulosa cells, and the hyaluronic acid will be released in the antral cavity in the fluid. And normally the hyaluronic acid will lead to water retention. And it has the property of retaining water, hyaluronic acid. So its presence in the follicular fluid will lead to more water retention in this enteral cavity, and thus we will have more water in this enteral cavity, and it can expand, it can increase in size, okay, and the antrum will increase in size importantly. And also under the action of the LH, or as a consequence of this LH surge, we will have a loosening of the intracellular matrix between the cortical layers of the granulosa cells and the peripheral layers, the mural granulosa cells, okay? We will have a loosening of the intercellular matrix between them. And this will help in the ovulation and an increase in the total blood flow of the follicle. So we will have an increase in the blood flow reaching the follicle because we will have, in fact, more blood vessels that will arrive to this follicle, to the theca. We have said that the theca is vascularized, and the theca externa, and also the theca interna. So we will have more blood vessel reaching this Graafian follicle or pre-ovulatory follicle. And as a consequence of those high levels or of this high blood flow reaching the Graafian follicle, the connective tissue surrounding the follicle will become hyperemic. In fact, those blood vessels reaching this theca will have some like pores in their walls, and the plasma can be a little bit, can diffuse a little bit to the theca, and so this theca will be will contain more water, okay, due to the plasma coming from the blood, and we can say that the connective tissue surrounding the Graafian follicle will become hyperemic, يعني like forming an edema, okay, يعني مثل انتفاخ في مي، يعني rich in water بصير هيدا التشو بصير rich in water. We will have a follicular edema. So we can say that the connective tissue will becomes hyperemic, okay? And the hyaluronic acid inside of the antrum, here we have the antrum, increases the water retention. Now, under the action of LH, the granulosa cells will produce hyaluronic acid and will release it in the enteral cavity. And this hyaluronic acid has the property of water retention, so it can bind water molecules. And since we have the hyaluronic acid in high concentrations now in the enteral cavity, it can bind more water molecules, and this enteral, the volume of the enteral fluid, because we have more water now inside of it, will increase in size, the volume will increase, and thus the enteral cavity will increase in size. And the increase in the size of this enteral cavity will lead to if you remember, we had several layers of granulosa cells, and now due to this increase in the size of this enteral cavity, this will lead to that we will only have one granulosa layer or one layer of mural granulosa cells, and the other cells will degenerate, okay? You can see here that here we had several layers of granulosa, and this is the enteral cavity. Now, once we have the expansion of this enteral cavity and the important increase in its volume, and this is the Graafian follicle or the pre-ovulatory follicle. Okay, so the increase in the size of this antrum will lead that we will have one layer of granulosa cells adjacent to the basement membrane, but the other layers will, in fact, the other cells will die. and one layer of granulosa cells will be kept. And this layer of granulosa cells is called the mural granulosa layer. Okay. And we will only have, we will keep on having only a small amount of granulosa cells surrounding the oocyte. Okay, and this sort of granulosa cells forming a stalk connecting the corona radiata, those layers of granulosa, to the mural granulosa cells. Okay. So we were talking about the increase in the volume of the antrum or the enteral cavity. This results in a relatively thin peripheral layer of granulosa cells, okay, which is the mural layer. And we will have a basement membrane and thecal cells to which the oocyte with its associated cumulus cells is attached only by a tenous and thinning stalk of granulosa cells. So we will have this, the oocyte with its surrounding corona radiata cells attached to the mural granulosa cells and to connected with the theca, the basement membrane and the theca by the this thin stalk of the cumulus oophorus. Okay. Now, the increasing size of the follicle and its position in the ovarian cortex, as you know, we have said that all the developing follicles, all the follicles in fact, are situated in the cortical region of the ovary. So if this is the ovary, the follicles are adjacent to the periphery of the ovary in the cortical region. Okay, so once this follicle will increase in size, this pre-ovulatory follicle has an important size now. It will bulge, it will protrude a little bit from on the surface of the ovary, okay, forming a stigma. Okay. So the increasing size of the follicle and its position in the ovarian cortex, because it has a peripheral position, this will cause this follicle to bulge from the ovarian surface, يعني بيطلع شوي, forming a stigma. Okay. Now the follicular wall will become thinner and avascular. The walls here will become thinner and avascular, because as you are going to see, we will have some proteases that will come and digest the extracellular matrix between the theca cells. Okay. And we have also the connective tissue breaks down and the follicle will rupture. We have also a rupture of the connective tissue at the levels of the ovary and of the peripheral, of the ovarian epithelium and of the tunica albuginea surrounding or adjacent to this pre-ovulatory follicle. So the tunica albuginea of the ovary and the epithelium of the ovary adjacent to this pre-ovulatory follicle and to the stigma will be digested by the action of enzymes. And so the connective tunica albuginea is composed from a connective tissue. So the connective tissue will break down, and also the connective tissue between the theca cells adjacent to this ovarian cortex. يعني the connective tissue between the theca cells here adjacent to the periphery to the stigma will be digested, and in fact this will help in the ovulation. Okay. When they are digested, the oocyte can be released. We will not have any membranes anymore here, and with the action of the muscular cells helping in the contraction, this will help the oocyte to be expulsed into the fallopian tube. Okay, and the follicular fluid flows out. Now the connective tissue break down and the follicle will rupture, releasing in this way the oocyte surrounded by the corona radiata cells. Okay, and the follicular fluid flows out, carrying with it the oocyte. So once we will have the ovulation or this follicular rupture, we said that we have also muscular cells, smooth muscle cells, some smooth muscle cells in the theca interna. And we have a myofibroblast in the theca externa. Okay, so those cells will contract under the action of prostaglandins, and due to their contraction, this will help the oocyte and the fluid to exit the ovary and to go in the fallopian tube. So the follicular fluid will flows out during the ovulation, carrying with it the oocyte and its surrounding mass of cumulus cells into the fimbriae of the uterine tubes or of the fallopian tubes that moved closer to the ovaries. So the fallopian tubes will move closer to the ovaries in response to estrogen during the pre-ovulatory phase, and they can now receive the oocyte. Okay. Is it clear till now?
Yes, doctor.
Now, how do we have the follicular rupture? We were talking about the action of, I was talking a little bit in general, and we had enzymes that will digest this ovary, the tunica albuginea, and a rupture of the ovarian epithelium, plus a little bit the connective tissue between the theca cells. Now here we will see more in details what are the enzymes, what related to this follicular rupture or ovarian rupture. We have several proteolytic enzymes. So proteolytic comes from protein and lysis. So they are enzymes related to having a lytic activity. They will lysate something. Okay, they will lead to the lysis of a tissue. Okay. Now, those enzymes are the matrix metalloproteinases, some matrix metalloproteinases. We have some serine proteases. We will talk about them. We have the prostaglandins, and we have the progesterone. Okay, the progesterone is not an enzyme, but it will induce the production of enzymes. Okay. And the prostaglandins also, they are not enzymes, but they will stimulate the production of some enzymes. So we have the rupture of the follicle is dependent on several proteolytic enzymes like the MMPs or the matrix metalloproteinases. In fact, those enzymes, they degrade the matrix, okay? And in this family of, these are a family of proteolytic enzymes containing the collagenase, okay? So the collagenase is a matrix metalloproteinase, and this enzyme, the collagenase, will be produced by the granulosa cells, and it will dissociate the cells in the area of the stigma. So the collagenase produced will dissociate the cells in the area of the stigma. So in the area surrounding the stigma and in the area of the stigma here, to digest this area and to let the oocyte exit the follicle and the ovary. We have the serine proteases also, like the plasmin and the plasminogen activator. In fact, both of them, they are proteases belonging to the family of serine proteases. The plasminogen activator, in fact, activate the conversion of the plasminogen into plasmin, And the plasmin will activate the conversion of inactive collagenase into inactive collagenase. And so we can say that the plasminogen activator will activate collagenase and the plasmin. And the plasmin and the collagenase will lead to the dissolution of the connective tissue in the area of the stigma. Okay, so now we have also the plasmin and plasminogen activator that are induced, and their production will lead to the activation of the collagenase and to the dissolution of the connective tissue in the area of the stigma. Here we can see that the plasminogen, it comes from the blood circulation. It will be under the action of the plasminogen activator will be transformed into plasmin, and the plasmin will lead to the activation of the collagenase, and it will lead to the conversion of the inactive collagenase into an inactive collagenase, and the collagenase will digest the collagen. Okay, the collagenase is the protease. It's a protease that will digest the collagen. That's why it's called collagenase. And as you know, the extracellular matrix is rich in collagen, specifically at the level of the tunica albuginea of the ovary. So this collagenase, by digesting, by breaking those collagen inside of the tunica albuginea and the extracellular matrix in the theca surrounding the stigma, will lead to the disruption of this tissue, and in this way this tissue will be degraded and the oocyte can exit the follicle and the ovary. Also we have we said that we have production of prostaglandins by the granulosa cells under the action of LH, and those prostaglandins are the PGE2 and the PGF2 alpha. And they have important role in the process of ovulation. In fact, the PGE2 stimulates the plasminogen activator that, as we have said, this plasminogen activator will activate the transformation of the plasminogen into plasmin, and the plasmin will activate the collagenase. Okay, so the PGE2 will stimulate the plasminogen activator known to activate collagenase and the plasmin. And the PGF2 alpha, which is the prostaglandin F2 alpha, called also the luteolytic factor prostaglandin F2 alpha. It's called luteolytic factor because it is responsible for the degeneration of the corpus luteum that will be formed later. We will talk about it. And that's why we call it the luteolytic factor, prostaglandin F2 alpha. The PGF2 alpha stimulates the rupture of lysosome in the apex epithelium, destroying the tunica albuginea and the theca. So the PGF2 alpha is secreted by those granulosa cells. Okay, it is produced and it acts in a paracrine manner on the epithelial cells of the ovary in the region adjacent to the stigma. And they have a local action adjacent to their place of production. They are produced by the granulosa cells, and they will act on the epithelial cells here in the region next of the stigma and adjacent to the stigma. And in fact, this PGF2 alpha will lead to the rupture of the lysosomes inside of the epithelial cells of the ovary and of the ovarian cortex. So if the epithelial cells, if the lysosomes will be ruptured now inside of the epithelial cells of the ovary, the lysosomes contains enzymes. So once they are ruptured due to the action of the PGF2 alpha, they will release their enzymes in the epithelial cells, and this will lead to the destruction of the apex epithelium or the cortex, the epithelial cortex, and to the destruction of the tunica albuginea in the region adjacent to the stigma, and also to the theca. Okay. So this will lead to the destruction of the tunica albuginea and the layer of epithelial cells in the stigma region and adjacent to it, and also to the theca, in the region next to this ovarian, next to the stigma, and to the ovarian cortex. So the PGF2 alpha also stimulate ovarian contractions, leading to the follicular rupture and follicular contraction for actual oocyte expansion. If you remember, we have talked in the chapter 2 about the action of prostaglandins, and also in the male reproductive system, we have said that we will have production of prostaglandins that will lead to contractions of the smooth muscles. Here also we have this prostaglandin F2 alpha that can act on the theca cells, because in the theca interna we have smooth muscle cells. We have also glandular cells in the theca interna, but we have some smooth muscle cells, and the theca externa is very rich in myofibroblasts that are like the smooth muscle cells. And so the myofibroblasts of the theca externa and the smooth muscle cells in the theca interna can respond to the action of this PGF2 alpha and contract, and this will lead to the expulsion. And due to their contraction, we'll have an expulsion of the oocyte outside of the ovary with the follicular fluid and with the corona radiata cells surrounding the oocyte. Okay, so the PGF2 alpha, from one side, they stimulate the rupture of the lysosomes in the cortical epithelium, destroying in this way the tunica albuginea and the theca, okay, and also the epithelial cells, and it stimulates also the ovarian contractions due to the contractions of the cells in the theca layers, and leading to follicular rupture and to follicle contraction and follicular rupture for actual oocyte expulsion. We have the progesterone. The progesterone will stimulate the production of collagenase by the theca cells, and this collagenase produced by the theca cells will lead to the dissociation of the extracellular matrix between the cells in the area of the stigma. Okay, so all of we can see here that we have we have talked mostly about the collagenase and the plasmin, the plasminogen activator and the plasmin that stimulates the digestion of the extracellular matrix. The prostaglandins produced by the granulosa cells will lead to also an activation of the plasminogen activator, and thus to the production, to an activation of the collagenase. The PGF2 alpha will lead to the rupture of lysosomes in the epithelium, in the cortical epithelium, and thus it will help also in the destruction of the tunica albuginea and the theca. And the PGF2 alpha will stimulate also the ovarian contraction by acting on the muscular cells or the smooth muscles and myofibroblasts of the theca. The progesterone will lead to stimulates the production of collagenase by the granulosa cells by the theca cells, and this will lead to the dissociation of the extracellular matrix at the level of the stigma. Okay, so from this point, at the level of the stigma, the oocyte will exit the ovary into the fallopian tubes. We can see here that we have, due to this LH surge, we will have the cells, we will have the granulosa cells that will be organized in a few layers surrounding the oocyte, forming the cumulus oophorus. Okay, and in this way the oocyte will have a big enteral cavity, and in this way the oocyte will be separated from the follicular wall. Okay, we will have under the action of this LH, as a consequence of this LH surge, we will have some protein synthesis and enzyme synthesis around the follicle and in the stigma region. In fact, we will have a prostaglandin, a synthesis of prostaglandins that will lead to the contraction of smooth muscles, and we will have also the production of plasminogen activator that will lead to the conversion of the plasminogen into plasmin, and the plasmin will lead to the activation of the collagenase. We will have production of progesterone under this LH surge, or under the action of LH, production of progesterone, which will lead to the production of collagenase by the theca cells. Okay. And all of those events will lead to the weakening of the follicular wall. So to the weakening of the basement membrane surrounding the follicle, to the weakening of the ovarian cortex at the level of the stigma, and also to the theca, to the dissociation of the theca cells surrounding at the level adjacent to the stigma, and this will help in the ovulation also. Okay. We will have also, due to this LH surge, an increased blood flow reaching the follicle, and we will have vascular permeability. And due to this vascular permeability, we will have more plasminogen reaching the follicles, and that's why it will be also, we will have a good concentration of plasminogen to be converted into plasmin under the action of plasminogen activator, and this will lead to the activation of the collagenase. Okay. We will have the follicular edema also, as we have said, like water retention surrounding the ovary that will lead to a follicular pressure. Okay, but mostly the ovulation will due to the weakening of the follicular wall and the contraction of the smooth muscle cells. Now, what are the changes at the level of the oocyte? We were talking about the changes at the levels of the follicle. Maybe I have missed a point. Okay, no, we have talked about all of these details. Now we will talk about the modifications at the levels of the oocyte. As we have said during the last session, the LH surge will lead to the resumption of meiosis. If you remember, we said that this is the oocyte, and those are the granulosa cells surrounding it. And here we have in blue the zona pellucida. We have gap junctions between the granulosa cells and the oocyte. And those, in fact, gap junctions can be maintained under the action of the OMI, of the oocyte maturation inhibitor produced by the granulosa cells. If this oocyte, this OMI factor or this oocyte maturation inhibitor levels are decreased, we will have closure of those gap junctions and disruption of those gap junctions. Now, in the absence of LH, before the LH surge, we had high levels of cyclic AMP inside of the oocyte. And those high levels of cyclic AMP were maintained, will lead to the inactivation of a factor called the maturation promoting factor. This factor can, when it is inactive, we will have an arrest of the cells in the meiosis 1, in the prophase, and more specifically in the prophase of the meiosis 1. And in fact, those high levels of cyclic AMP were maintained high because we had some cyclic AMP where levels are coming from the granulosa cells through the gap junctions, and also we had the cyclic GMP, which was produced by the granulosa cells and also entering the oocyte through the gap junctions. And the cyclic GMP, as we have said, they inhibit the hydrolysis of the cyclic AMP. So as long as we have cyclic GMP coming inside of the oocyte, the cyclic AMP levels will be maintained high and the MPF will be inactivated, okay? However, and then we will have a meiotic arrest in the prophase 1. Now, in the case where we have a surge of LH, what will happen? We have said that this LH surge will lead to a decrease in the OMI levels, and this decrease in the OMI levels will lead to the closure or the disruption of those gap junctions between the granulosa cells and the oocyte, and thus the cyclic GMP can no more enter inside of the oocyte. So the cyclic AMP inside of the oocyte can be hydrolyzed or degraded, and as a consequence of its hydrolysis, we will have reduced cyclic AMP levels, and the MPF will be activated, and as a consequence of the MPF activation, the meiosis resumes and the meiosis 1 resume. Okay, in fact, this MPF activation will lead to the degradation of the to the chromosomes condensation and the degradation of the nuclear envelope during the prophase, and this will help the cells to exit the prophase. Okay, and thus the meiosis will resume during this MPF activation. So here we can see that as a consequence of the LH surge, we can have on the oocyte, we can have a resumption of the meiosis 1. So due to the LH surge, we will have a decrease of OMI levels and closure of gap junctions, and this will make that no more transfer of cyclic GMP to the oocyte is made, and this will lead to a decrease in the cyclic AMP levels. And as a consequence of the decrease in the cyclic AMP levels, the nuclear membrane will break down, and we will have the arrested meiotic prophase is ended many years after its initiation. So the cells that were arrested in their meiosis, in the prophase 1 of the meiosis, will end their this arrest, and they can resume, they can continue this meiosis 1 many years after its initiation, because we said that the females will be born with the oocyte arrested in the prophase of the meiosis 1. And the meiosis 1 will be achieved and the cells will start the meiosis 2. So as a consequence of the meiosis 1, we can have the production of the secondary oocyte and one polar body. The secondary oocyte will enter directly in the meiosis 2. And during the meiosis 2, we have the meiosis 2 starts, and the chromosomes will lie on the second metaphase spindle. However, the meiosis 2, this meiosis will be arrested in the metaphase stage, and the oocyte will be ovulated while it is arrested in the metaphase 2. Okay. When we have the ovulation process, the oocyte will be ovulated as arrested in at the metaphase 2, and it will remain arrested at the metaphase 2 until the fertilization. If the fertilization will occur, this meiosis 2 will resume. If there is no fertilization, the meiosis 2 will never be achieved. Okay, now what regulates also this arrest of the meiosis 2 and how the cells can exit due to the fertilization, how the cells will exit this metaphase 2. This arrest in the metaphase 2 is due to the presence also of the MPF factor. We talked about the MPF here in the meiosis 1, and we will see its role in the meiosis 2 also. This MPF factor is composed from two subunits. So from two proteins, okay, the cyclin B and the cyclin dependent kinase 1. And the arrest in the metaphase 2 is also controlled by the cytostatic factor called CSF. The CSF factor is also composed from two proteins, which are the CMOS and the cyclin-dependent kinase 2. Now, in the meiosis 2, we have the CSF, and the CSF stabilizes the MPF. As long as this MPF is stable, the cells will be arrested in the metaphase 2. Once this MPF is destroyed or deactivated, the meiosis can resume their metaphase 2. Okay, so the arrest in the metaphase 2 is under the dependence, or is dependent from the MPF and the CSF. Whenever the MPF is present, we have the MPF composed from two subunits, the cyclin-dependent kinase 1 and the cyclin B. Both of them form the MPF. The presence of the MPF now in the meiosis 2 will lead to the stabilization of the spindle, of the meiotic spindle, or of the microtubule spindle, and thus to the arrest of the cells in the metaphase 2. And this MPF is made stable due to the action of the CSF. So whenever we have the CSF, the MPF is stabilized. If there is no CSF, we'll see how now, the MPF will be no more stable. So we will have a dissociation of the complex, and the microtubule spindle will no more be kept stabilized, and the cell can exit the metaphase 2 to continue the meiosis 2. Okay, and now after the when we have the fertilization process will lead to an increase of the intracellular calcium levels. So this will lead to the increase of the levels of calcium inside of the oocyte 2. And this increase in the calcium levels will lead to the destruction of the CSF. And this will lead to the dissociation of the MPF factor, or not exact, the dissociation of the MPF factor. And as a consequence of the dissociation of the MPF factor, the microtubule spindle will no more be stabilized, and the cells can exit the metaphase 2 to start the anaphase, and thus they can resume the meiosis 2. Okay, so here, as a consequence of the fertilization, we will have a rise in the calcium, that in the intracellular calcium inside of the oocyte 2. This will lead to the destruction of the CSF factor. As a consequence of the destruction of the CSF, we will have a release and activation of a factor called the anaphase promoting factor, or APF. This APF will lead to the ubiquitination of the cyclin B. The cyclin B is the subunit, one of the MPF subunits. So once a protein is ubiquitinated, the ubiquitination is a signal, that means is a signal to for the degradation of this protein. So whenever the cyclin B is ubiquitinated, it will be degraded. Okay, so we will have the proteolysis and destruction of the cyclin B. And if there is no more cyclin B, the MPF will not find enough cyclin B to form the complex. Okay, and thus we will no more have MPF, and the cells can exit the metaphase 2. Okay, so as a consequence of the proteolysis of the cyclin B, there will be not enough cyclin B to complex with the cyclin-dependent kinase 1, which is the other subunit of the MPF factor. And thus we will have no more MPF complex. Okay, and in the absence of the MPF complex, the cells can exit the meiosis, the metaphase 2, and the meiosis 2 can progress, and will be resumed. And this is why, as a consequence of the fertilization, the cells can exit the metaphase 2, and this is why it's only after the fertilization that the cells can resume the meiosis 2 blocked in the metaphase 2. In fact, because we will have a rise in the intracellular calcium, destruction of the CSF. This CSF normally stabilizes the MPF complex. When it is destroyed, the MPF complex will be destroyed and the meiosis can progress. And in fact, the CSF, it will lead to the release of the anaphase promoting factor. This APF will lead to the proteolysis of the cyclin B or degradation of the cyclin B, and thus there will be no more the MPF complex will not be able to form, and the meiosis can progress. Now, sometimes it can be a little bit it can create confusion about the role of MPF, because as we have said here in the meiosis 1, when it is inactivated, the meiosis 1 is arrested, and when it is activated, the meiosis 1 resumes. Here in the meiosis 2, in fact, when this complex is inactivated, the meiosis 2 can resume. Okay, so it is the opposite. Why? Because this MPF has different substrates. So it has different targets in the cells. From one side, it leads to the degradation of the nuclear envelope and the chromatin condensation. And thus it will lead to the exit of the cells from the prophase, because we need, when the meiosis starts, we need this degradation of the nuclear envelope and the chromatin condensation in order to proceed in the meiosis 1, to end this prophase 1 and to proceed in the meiosis 1. So when this MPF is inactive, we can't have the degradation of the nuclear envelope and the chromatin condensation, and that's why the cells will be blocked in the prophase 1. Okay, so whenever this MPF is inactive, during the meiosis 1, the cells will be blocked at the prophase 1. However, this MPF affects the microtubule spindle and the chromosomes assembly on the spindle. And that's why during the meiosis 2, in fact, so that's why it will lead to the stabilization of this mitotic spindle. And during the meiosis 2 and the metaphase 2, when the cells enter the metaphase 2, if this MPF is active all the time, the cells, this mitotic spindle, or let's say the microtubule spindle, will be kept stabilized and the cells will remain blocked in the metaphase 2. So we need to disrupt this MPF factor, we need to destroy it in order to proceed in the meiosis 2. Okay, is it clear for you? Are you still with me?
Yes, doctor.
Okay. وهنا مش مطلوب منكم to study the roles of MPF, but this is only to clarify why it has distinct roles in the meiosis 1 and in the meiosis 2. Okay, so in the meiosis 1, it is once we have the cells arrested in the meiosis 1, the MPF is inactive. We need to activate it to resume the meiosis 1. In the meiosis 2, the cells are blocked. Whenever this MPF is present and active, the cells remain blocked in the metaphase 2. We need to disrupt this MPF complex in order to proceed in the meiosis 2. Okay. Okay, what time is it? I will continue just for 10 minutes and then we will continue next week. Now, we have also we talked about the meiosis at the level of the oocyte that under the action of the LH, it will exit the meiosis 1, start the meiosis 2, but will be blocked at the metaphase 2, and it will remain blocked under the fertilization. Now also, we have other changes at the level of the oocyte. We have a cytoplasmic maturation, where the contact between the oocyte and the granulosa cells of the cumulus oophorus will be broken. So the gap junctions, in fact, between the granulosa cells and the oocyte will be broken following this LH surge by withdrawal of the cytoplasmic processes that were traversing the zona pellucida. Also, we have said here that under the action of LH, we have an increase in the OMI levels in the granulosa cells, and this will lead to the disruption of the gap junctions. Okay, gap junctions. So this is how the LH surge will lead to the disruption of those gap junctions or connections between the granulosa cells and the oocyte. And also we will not have a cytoplasmic, those cytoplasmic extensions or processes that were traversing the zona pellucida of the granulosa cells, of the cumulus oophorus cells will be disrupted also. Okay. And the Golgi apparatus in the oocytes will synthesize lysosomal-like granules. So the Golgi apparatus will synthesize granules containing enzymes. We call them here lysosomal-like granules that will be called the cortical granules. In fact, those lysosomal-like granules will migrate toward the cytoplasmic membrane of the oocyte, and they will remain below the cytoplasmic membrane and under the cytoplasmic membrane. Okay, and that's why. So they will assume a subcortical position. Cortical يعني periphery, يعني they will have a subcortical position. They will be under the surface of the ovary, sorry, of the oocyte. And that's why we will call those lysosomal-like granules cortical granules. And those cortical granules, in fact, they will be needed during the fertilization process to avoid the polyspermy. The polyspermy, yani to avoid that more than one sperm cell will fertilize the oocyte. Okay, so we need those cortical granules to avoid the polyspermy process. In fact, after that, the first sperm cell will reach the zona pellucida of the oocyte and will interact with it. Those cortical granules will be released, and they will lead to a thickening of this zona pellucida, and thus in this way they will prevent the interaction of the other sperm cells with the zona pellucida and consequently with the oocyte. Okay, so during the LH surge we will have the synthesis of those cortical granules or the release of those granules by the Golgi apparatus, and they will migrate toward the surface of the oocyte, and they will have a subcortical position. They will be positioned under the membrane, and they will be called cortical granules. Also, the centrioles will be lost in the oocyte, and only the pericentriolar material will be kept. Okay, and only the material surrounding the centrosomes will be kept to organize the microtubules during the zygote division. So we have said that the sperm cell will keep a centriole, will have the centrioles, and the pericentriole materials will be provided by the oocyte. Okay, and they will be used for the zygote division. And this oocyte will have a protein synthetic activity that will continue inside of this oocyte at the same rate, but now under the action of LH, new and distinctive proteins will be synthesized. This activity prepares the oocyte for fertilization. Okay, so we will have new proteins that will be synthesized to prepare the oocyte for the fertilization. We will talk about the corpus luteum in the next session. Okay, so for today, this is all for today. Do you have questions?
دكتور يعطيك العافية.
الله يعافيكم.